After getting to know the captivating expedition of Victor Ambrose and Gary Ruvkun on microRNAs, let us now delve into another such mind stimulating course. As SKA embarks on the path of discussing the recent Nobel Awards, here he takes us into the details of how three great minds got together from worlds apart into giving science a tool that is changing the course of treatment. David Baker, Denise Hazzabis, and John Jumper's story is a beautiful amalgamation of academics and industry. Of what came out is being witnessed by the world and has been fairly honored by the Nobel Prize. Here is their achievement, acknowledged and discussed rightfully so by SKA. Keep subscribing, sharing, and letting us know of your views for better podcasts ahead. Thank you and enjoy this episode.
Hello, my friends. Hope you are doing all well. Thank you for listening to this podcast and welcome back once again to Immunology for Everyone. As students of science, we learned that the diversity of life testifies the amazing capacity of proteins as chemical tools. It is important to know that all the chemical reactions that are the basis of life are driven by proteins only. This is because most of the functional molecules in all the metabolic reactions in our body that include enzymes, hormones, signal substances, antibodies, or even the building blocks of different tissues are proteins in nature. So let's think about on how the proteins perform their diverse functions. Proteins, as we know, are the polypeptide chains of different amino acids which are held together with covalent bonds which are called the peptide bonds. And like a DNA molecule, which is actually a chain of four nucleotides which are held together, and the uniqueness of a gene lies in the sequence of these four nucleotides. Whereas the uniqueness of a protein lies in the sequence of uh, 20 amino acids in general. So the sequence of amino acids, in fact, cause the folding of each polypeptide chain, giving it a unique secondary structure, which carries the alpha helix or beta pleated or a combination of these two chemical forms. And then the polypeptide chains, they fold further because of many different bonds which are hydrogen bonds, ionic bonds, the disulfide bonds between various amino acids, and that gives rise to a tertiary structure, which is a globular protein. And that tertiary structure is of the same polypeptide chain, which become takes up a specific structure of that particular protein. And amazingly, you would like to know that there are some proteins in nature which remain functional only when there are more than one units of different polypeptide chains are held together in the same molecule, and that basically makes it a quaternary structure of the protein where there can be more than one globular protein units are present. The very good examples of this are the hemoglobin, which has got four units which are held together with the help of some weaker ionic bonds like hydrogen bond and London dispersion forces. And we also know about the DNA polymerase, which also has got four units in the same molecule, and that makes them functional. So, and this folding and formation of tertiary structure of proteins is very important for the functioning of that particular protein. It is so important that any misfolding or no folding leads to non-functional state, not only the non-functional state, but aggregated form of the protein, which can also deposit in form of uh, misfolded protein, and that can cause many disease conditions due to non-functioning and deposition of that protein. And there are many examples of such diseases like Alzheimer's disease, Parkinson's disease. They are all caused because of the misfolding of basic proteins. And take the example of type 2 diabetes, which is caused by misfolding of islet amyloid polypeptide. And more examples are prion disease, cystic fibrosis, sickle cell anemia, muscular dystrophy, etc., etc. And unfortunately, there are currently no cures for protein misfolding. We can actually slow it down, but once the misfolding occurs, the aggregation occurs, they become very difficult to remove from the system. So, what does that indicate? This all indicates that the final folded structure of any protein is so important for its proper functional state. And it is obvious that how a protein is folded is determined by its amino acid sequence. But do you know that even a small protein of only 100 amino acids can have, 10 to power 47 possible three-dimensional structures? So predicting a protein structure from its amino acid sequence became a key problem in molecular biology. And whether the final structure of any protein can be predicted or designed so as to make any modifications to make it function as desired was again another curiosity with the molecular biologists and immunologists together. So, in this context, there are actually two related discoveries which have been recognized by the Royal Swedish Academy of Sciences this year, and that led to the award of Nobel Prize in Chemistry for the year 2024. One of the discoveries concerns with the construction of amazing novel proteins by computational designing, while the other is about predicting the protein structure from their amino acid sequences. And so both of these discoveries have opened up vast possibilities in this direction. And that's why I decided to discuss this story in this podcast today. So let's talk about these amazing discoveries one by one. The first discovery of how to design the functional proteins using computational tools was made by an American scientist named Davis Becker, who gets the one half of the total prize money for his amazing contribution to computational protein designing. Incidentally, he's a professor of biochemistry, Howard Hughes Medical Institute investigator, and director of the Institute of Protein Design at the University of Washington School of Medicine at Seattle in USA. And his research combines computational and laboratory techniques to create custom proteins with a very high precision. So Baker led a consortium of researchers and labs, which are known as Rosetta Commons, that pioneered the field of developing world's most popular and effective software for computational protein design, and succeeded in creating the first protein with a novel 3D shape, which was an inert molecule and called the top 7. And since then, the laboratory has produced several thousands of new proteins, including molecules that neutralize the viruses, target cancer cells, and even serve as catalysts in the chemical reactions. And in 2021, his team reported a very nice article in Journal Science uh, that is about the development and initial applications of a software tool which is known as Rosetta Fold. And that uses deep learning to quickly and accurately predict the protein structures based on very limited information. And it is amazing to know that with the Rosetta fold, a protein structure can be computed in as little time as only 10 minutes on a single gaming computer, and without which it can take sometimes years of a laboratory work to determine the structure of just one protein. This software, Rosetta Fold, for your information, is a three-track neural network, which means that it simultaneously considers patterns of protein sequences, how a protein's amino acid interact with one another, and the protein's possible three-dimensional structure. So in this architecture, one-dimensional, two-dimensional, as well as three-dimensional information flows back and forth, which allows the network to collectively reason about the relationship between protein's chemical parts and its folded structure. So this amazing software has since been used to compute hundreds of new protein structures, which includes many poorly understood proteins from the human genome also. And Baker's team has also generated structures directly relevant to human health, including for proteins associated with problematic lipid metabolism, inflammation disorders, as well as cancer cell growth. And it is really heartening to find that this Rosetta Fold code is available through a GitHub website, and anybody can submit novel or even artificial proteins to this website server for prediction of structure. This way, his work has helped revolutionize the field, making it possible to custom build proteins to meet many researchers specifications. So that is amazing discovery. Now let's talk about the second discovery, which is equally interesting. The second discovery also concerns the prediction of protein structures from the sequence of amino acids with a tool which is based on artificial intelligence, which is a step further. Since 1970s, the researchers had tried to predict protein structures from amino acid sequences, but this has been a notoriously very difficult task. However, 4 years ago in 2020, there was a stunning breakthrough. And That's when two researchers, Demis Hassabis, who's a computer scientist, and John Jumper, who is also a programmer, presented an artificial intelligence model for predicting the structure of any unknown protein. And interestingly enough, Demis Hassabis is only 48 years old. He was born in 1976 in London. And John Jumper is even younger. He is only 39 years old and was born in 1985 in US. And it is an interesting story how Demis and John they got into this discovery, and I'll relate a part of that. Demis Hassabis was the eldest son of Greek Cypriot father and a Chinese Singaporean mother. Uh, he was born in London. He developed very keen interest in chess and computer programming very early in his life, as he spent his life in those areas. So, as a chess player, from the age of 4 only, he obtained the rank of chess master at the age of 13. So you can imagine how curious he was, and he was playing so nicely. The chess game. So his affection for computer games inspired him to teach himself computer programming from books, and he uh, he wrote the code of his first game, which is a version of Othello at the age of only 17. And then he became so well versed with programming that he started working for a computer games company, which was called Bullfrog Productions, where he served as the lead developer at that young age of 17 of AI generated video game. Very famous, you might have heard about the theme park in 1994. So he was the main, lead AI developer of the AI generated video game. He actually took formal education as a computer science from the University of Cambridge, and but there also he kept playing chess, and he was champion in one of the sessions when he was ranked as the top chess player in the university. But after graduating from Cambridge, he worked as a lead AI programmer in gaming studios and developed many new AI games, including Black and White in 2001, Republic the Revolution in 2003, and Evil Genius in 2004. So basically, what I'm trying to say is that this guy was into computer gaming and programming, and he was developing AI based gaming platforms. But later, uh, he took his passion of AI algorithms in a different direction because he pursued his PhD in cognitive neuroscience at University College of London with the topic to understand how the human brain managed imagination and memory. So after learning that, after graduating and his doctoral degree, he did his postdoctoral work in Harvard University and Massachusetts Institute of Technology, and in similar subject, and he became the Henry Wellcome Fellow at University College of London once again. And later, when he was in London, Demis collaborated with a New Zealand computer scientist, Shane Lack, and a English, entrepreneur, Mustafa Suleiman, to set up his AI startup company, which he named as DeepMind, that he set up in 2011. And there, in DeepMind, he developed a series of AI models and trained them using deep learning to play and master video games. So now he sold his DeepMind company to Google, but they appointed him as the CEO of the company, which was now called DeepMind Google, platform. So at DeepMind um, uh, AI company, Demis developed many game programs like AlphaGo and AlphaGo Zero that had a neural network that learned to play Go game from human players. So that basically improved the programming in a better way. And that then Demis became interested in applying what he learned from those AI successes to scientific problems. So it is very interesting to know that in 1994, when the curiosity of many molecular biologists and basic scientists was in knowing how the protein structure can be predicted, there were two biologists, John Malt and Christoph Fidelis, who started a contest basically, which was named as Critical Assessment of Protein Structure Prediction Challenge. And they were trying to test the methods for predicting protein structures. So it was a sort of contest, and every 2 years the contestants were given the amino acid sequences of proteins whose structure had been determined but not published, and the contestants were challenged to predict the protein structures. And by the end of 2020s, most models that this challenge could gather were only up to 40% accurate, which was very, very low. So that's the time when Demis, with his DeepMind company, also entered into the protein structure contest, and they brought in their AlphaFold program in the contest, which was CASP 13 in 2018. And amazingly, they delivered an astonishing accuracy of about 60%, which was far ahead of any other competitor. However, they still want to improve beyond that, which was very difficult. But then a very interesting thing happened. The DeepMind hired another, programmer, John Jumper, who had an experience with protein simulation, and he joined the team and DeepMind, and then he led to development of another improved version of AlphaFold, which he called as AlphaFold 2 AI program. And then AlphaFold 2 program, which was trained on databases of amino acid sequences and protein structures, and used a neural network which was called as transformer. And that was used to find the likely protein structure. So amazingly, in the next contest, which was CASP 14 contest in 2020, the AlphaFold 2 reached an accuracy of 90%, which was basically comparable with the experimental results which the scientists had in their laboratory. So that indicated that the problem of finding a protein structure when the amino acid sequences are given was nearly solved. So Demis, John, and his collaborator, they used AlphaFold 2 to calculate the structure of more than 50,000 human proteins in 2021. But then they went further. They went to on to calculate the structures of almost all 200 million known proteins, which basically come from about 1 million different species which are living in this universe. And that's why Demis call it the entire protein universe. They have been able to predict the structure of 200 million known proteins. So, very, very astonishing. So, in summary, Demis described his research, and I quote that the computational biology has long held tremendous promise for creating practical insights that could be put to use in real world experiments. And AlphaFold 2 AI delivered on this promise. There are further a universe of new insights and scientific discoveries which are made possible by use of artificial intelligence as a scientific tool. And artificial intelligence, on the other hand, has long known incredible potential for use in scientific research, and AlphaFold is a proof of concept of that. So, as more scientists adopt AI for use in everything from building data to simulating experiments, drug design, modeling complexity, discovering novel solutions for extant problems, and building upon existing knowledge, uh, it will continue to see fundamental scientific breakthroughs in the years to come. So, ladies and gentlemen, now we know that the life could not exist without proteins, and that we can now predict protein structures and design our own proteins can be considered the greatest benefit to mankind. Among many scientific applications, researchers can now better understand antibiotic resistance and create images of enzymes that decompose even plastic. So there can be many such applications. And for a more living example, I may quote here a personal experience which helped us in my laboratory a few years back when one of my PhD students, Aryan, who used bioinformatic tools to map the T cell specific epitopes from some previously cloned genes of a parasite called Leishmania donomini, And while trying to make a multi-epitopic vaccine against the parasitic infection, he used some of such web tools available free of cost to predict the structure of a totally artificial protein. And that protein incidentally induced sterile immunity in the animal model against the lethal challenge that we did with the virulent strain of that parasite. So, friends, such discoveries from David Becker, Demis Hassabis, and John Jumper have made a visible change. The way researchers all over the world now can work with ease, even under resource limited conditions, to generate molecules that can be of immense importance, like vaccines or other drugs, not only for finding the answer to their research questions, but also in alleviating the human sufferings in general. So, thank you very much for listening today. And I hope to bring more interesting topics in subsequent episodes of this podcast. So, stay tuned, keep listening, and take care.